Welcome to the deep dive. This is where we take your sources and we really plunge into the essential knowledge you need.
Yeah, we try to cut through all the complexity, find those, surprising insights and the really clear takeaways.
And today we are diving deep, really deep into the very building blocks of life. Two molecules, absolute stars, DNA and RNA.
Right. They sound similar, maybe, but their differences, well, that's what makes everything in the cell actually work.
We've got a great source for this, an article that really lays out the key distinctions between them.
Think of it like a guide to how your cells divide up the really important jobs.
So our mission today, let's unpack that source. Let's highlight the essential ways DNA and RNA are different. Their chemistry, their shape.
Where they are in the cell, what they actually do, and why they're built so differently for those roles.
Yeah, it's all about understanding why life needs both of them.
It's a beautiful system. Specialization right down at the molecular level.
Okay, then, let's unpack this. So maybe the first place to start. What are they? I mean, the source points out the first big clue is, well, it's right there in the names, deoxyribonucleic acid versus ribonucleic acid.
Exactly. And that difference, deoxy versus just ribo, it tells you something crucial about the sugar that forms the backbone of each molecule.
DNA uses deoxyribose sugar. RNA uses plain ribose.
Right. And the source notes deoxyribose is just missing one single oxygen atom compared to ribo.
Just one oxygen. That sounds incredibly small, doesn't it?
It does sound tiny, but chemically speaking, phew. One atom can change, well, everything. That missing oxygen makes a massive difference in stability.
How so?
Think of it like removing a slightly weaker link in a chain. Without that extra, slightly more reactive oxygen, the DNA backbone is just much tougher, more resistant to chemical attack, things that could break it down.
Okay. So the very first difference we see, the sugar, it directly affects how robust the molecule is, how long it lasts.
Exactly. Which makes total sense if its job is storing critical information long term.
That's right, designed for permanence.
Yeah. Okay, so then there are the other building blocks,
Okay.
the nitrogenous bases. These are the letters A, G, C.
The genetic code letters. Both DNA and RNA use adenine, A, guanine, G, and cytosine, C. That part's the same.
But here's another key swap the source highlights. DNA has thymine, T,
And RNA doesn't use thymine, it uses uracil instead.
U. So you've got A, G, C, T for DNA and A, G, C, U for RNA.
That T versus U thing. It's like a little molecular tag, isn't it? Instantly tells the cell machinery, okay, I'm dealing with DNA, or, this is RNA.
Precisely. And it also affects how they pair up. In DNA, A always pairs with TC with G. That's the rule.
But in RNA.
In RNA, A pairs with U. C still pairs with G, though. That pairing is, well, it's fundamental for everything. Copying information, reading it out.
Okay, so we've got different sugars affecting stability, a different base acting as an identifier. What about their shape? Because the DNA double helix, that's iconic.
Oh, absolutely. It's like that twisted ladder, right? Two strands wound really tightly around each other. The source calls it a tightly associated pair.
And that double-stranded structure, it must add even more stability.
Definitely. Another whole layer of stability. Plus, it gives you a built-in backup copy. If one strand gets damaged, the cell can often use the other strand as a template to fix it.
Right, yeah. Tough, redundant. Okay, so what about RNA then? If DNA is the double helix.
RNA is usually single-stranded. It doesn't typically have that partner strand twisting around it.
Just one strand on its own.
Yeah. And the source mentions it exists as relatively short chains, much shorter than the huge DNA molecules you find in chromosomes.
A single DNA molecule from one of your cells, if you could unravel it, could be, centimeters long?
Okay.
It's massive.
Absolutely. Whereas RNA molecules are typically just, a few thousand bases long, maybe orders of magnitude shorter.
And being single-stranded, I guess that makes it less stable again, but maybe more flexible.
Exactly. Less rigid than the DNA double helix. It can fold into more complex shapes, actually, but it's generally less stable overall. A different form for a different function.
Okay. Different building blocks, different letters, different shapes, different lengths.
Yeah.
Influencing stability all the way.
Yeah.
So where do these things actually live and work inside the cell? They're not just floating around randomly, are they?
No, definitely not. In complex cells, like ours, eukaryotes, DNA's main home is the nucleus. Think of it as the secure vault, the command center.
Where the master blueprint is kept super safe.
Right. There are tiny bits elsewhere, like in mitochondria, but the vast, vast majority is protected inside the nucleus.
Okay, but RNA.
Yeah.
It's made in the nucleus, right? copied from the DNA.
Yes, it's transcribed from the DNA template there.
But it doesn't just stay there.
Nope, that's key. Different types of RNA molecules, once they're made, they actually move out of the nucleus into the cytoplasm.
The main body of the cell.
Yeah, the bustling factory floor, you could say. The DNA, that irreplaceable master copy, stays put, protected. RNA, the working copy, travels out to where the action is, where the proteins get built.
Which brings us nicely to function. What are they actually doing? The source paints a really clear picture here, that division of labor again.
It's quite distinct. DNA's main role, its big job, is information storage. Long-term storage, it's the archive.
The complete instruction manual for building and running?
Yeah.
Well, everything, the whole organism.
Exactly. It holds the code for making proteins, for directing how cells specialize, for heredity, passing information down through generations.
So it just sits there, relatively speaking, holding the plan, which means RNA must be the one actually using the plan, putting it into action.
That's a great way to put it. RNA is the go-between, the intermediary.
Okay.
It takes a specific instruction like, say, the recipe for one particular protein from that giant DNA blueprint in the nucleus,
Okay.
carries that specific message, that copy, out into the cytoplasm.
Right, onto the factory floor.
And then it actively helps build that protein. It acts as the messenger, sometimes the translator, even part of the machinery itself, the ribosome.
Ah, so it's doing multiple things.
Yeah.
The source says RNA is more versatile than DNA, capable of performing numerous, diverse tasks. It's not just one job.
Not at all. Messenger RNA carries the code, transfer RNA brings the amino acid building blocks, ribosomal RNA forms the core of the ribosome. It's involved in many steps.
And that versatility, that dynamic role,
Yeah.
does that connect back to the stability thing we talked about earlier?
Absolutely, directly. DNA stability, remember. From the deoxyribose sugar, the double helix structure, it's built for permanence, protection. It resists breaking down.
It's the critical hard drive. You really don't want that crashing.
Well, precisely. RNA, though, with its ribose sugar and single-stranded form, it's inherently less stable, more chemically reactive.
Okay.
The source points this out. It's not stable in alkaline conditions. Its structure makes it easier for enzymes in the cell to find it and chop it up.
But wait, why would you want your working copy, your messenger, to be less stable? Wouldn't you want that to be pretty robust, too? Seems counterintuitive.
But this is where it gets really clever. That instability, it's not a bug, it's a feature. It's part of the design.
How?
Well, the source says RNA strands are continually made, broken down, and reused. Think about it. The cell needs to make specific proteins based on what's happening right now. Maybe it needs a lot of one protein for a bit, then needs to stop making it and make something else entirely.
Oh, I see. If the messenger RNA for that first protein was super stable like DNA, it would just hang around forever, right? Keep making that protein even when the cell doesn't need it anymore. It would clog up the works.
Exactly. Its transient nature, the fact that it can be made quickly when needed and then broken down just as quickly when the job's done, makes the whole system incredibly responsive and adaptable.
Dynamic.
Very dynamic. It lets cells react fast to changes in their environment or signals they receive. DNA is the permanent library. RNA is more like a constantly updated set of Post-it notes or work orders.
That makes sense. And the source mentions another related point, proofreading. DNA gets proofread when it's copied to catch errors.
Yes, there are sophisticated repair mechanisms.
But the enzyme that makes RNA polymerase, it didn't really proofread much, does it? So RNA tends to have more errors, a higher mutation rate.
Which, again, kind of fits the model. An error in a temporary RNA message is less of a disaster than a permanent error etched into the DNA master blueprint.
Right. It's gone quickly anyway.
Yeah. It just highlights again which molecule is prioritized for that ultimate accuracy and long-term fidelity.
Okay, so stepping back, looking at the big picture, this whole list of differences, sugar, base, structure, stability, it's not just, biochemical trivia.
Not at all.
This division of labor, built on these fundamental chemical and structural differences, it's absolutely essential for life as we know it.
It really is. You need the stable, protected archive that's DNA. And you need the dynamic, versatile workforce to read the archive and build things that's RNA.
It's an incredibly elegant solution, isn't it? To manage this vast amount of information, keep it safe, but also use it effectively, moment by moment.
How do you store the encyclopedia safely, but also look up just one entry, make a quick copy, take it to the workshop, and translate it into a tool?
You invent DNA and RNA. Their distinct properties are just perfectly suited for their separate but totally interdependent roles.
The ultimate cellular tag team.
So let's quickly recap. We saw the sugar difference. Deoxyribose and DNA for stability, ribose and RNA for reactivity.
The key base swap. Thymine in DNA, uracil in RNA, acting like an identifier.
The structure. DNA's stable double helix versus RNA's more versatile, usually single strand.
Where they live, DNA mainly in the nucleus vault, RNA made there but working out in the cytoplasm factory.
Their core jobs. DNA as the master blueprint storage. RNA doing the dynamic transfer and building work.
And crucially, how their stability or lack thereof for RNA is perfectly tuned for those jobs. DNA built to last, RNA built to be transient.
These aren't just like random variations. They are the fundamental basis for how genetic information gets stored, accessed, and used in pretty much all life.
It's the core system.
So what does this all really mean? Where does this leave us?
Well, maybe think about this. We often focus on DNA's stability as being paramount, right? The unchanging blueprint. But consider how the fact that RNA is less stable, that it is more reactive, that it is constantly being made and broken down, maybe that's what actually makes it more versatile. More capable of doing all these different temporary jobs compared to the DNA that's locked away. Think about how that dynamic flexibility, that ability to be transient, to be recycled quickly, how that's just as vital for keeping life going, maybe even more vital moment to moment than the absolute stability of the archive itself.
So it's the interplay, the dance between the stable archive and the dynamic messenger.
Exactly. It's that constant dance between stability and dynamism that keeps the whole cellular factory humming along. That transient nature isn't a weakness. It's RNA's superpower.